Good afternoon, everyone. Myself, Pawan Kumar, Associate Professor in Department of Pharmaceutical Analysis. Today, I'm here to discuss about drug discovery and its stages. So, let me tell what is meant by drug discovery. It is defined as a process which identifies a compound which is therapeutically active in treating the diseases. So, drug discovery involves so many steps, which includes the identification, synthesis, characterization, screening, and assay for therapeutic efficacy. Before going for the drug discovery, it is most important for every pharmaceutical industry to develop the drug which is particularly suitable for the treatment with lesser side effects or minimum side effects and no adverse drug reactions. Let us see the few stages of drug discovery. The few stages includes the target identification, target validation, lead identification, lead optimization, pre-clinical safety, and the most important one, clinical trials, which plays a major role in the identification of the drug development stages. So let's see the first one, drug target identification. The target identification is the first and foremost step in the drug discovery channel because a drug target is the specific binding site for a drug through in which it will exert its therapeutic action. Usually, the drug target refers to a single biomolecule, and a specific drug targeting process involves the following characteristics, which includes maybe the drug can be a molecule like protein. This biomolecule should have that specific sites or locations for its mechanism of action and execution of action. The biomolecular structure may change when it binds to the drugs. The physiological response of the molecule plays a major role in regulation or complex function of the drug molecule. Hence, it is most important for the first step of drug discovery, which will be called as the target identification, which plays a major role in this scenario. And there are few limitations also for this, because the drug target should undergo the pathogenesis of disease, it should not disturb the body homeostasis, and the drug target relation should not be 1 to many. If one crosses this, the next step was target validation. It helps to develop the new drug and development research and provide more views on pathogenesis of the target related disease. The validation process includes discovery of biomolecule, evaluation of its potential as a target, designing a bioassay, constructing a high screening method, performing screening to find hits, and evaluation of hits. The lead discovery. It is most important for everyone to go for the lead discovery because it contains the compounds which are chemical, synthetic peptides, dyes, natural compounds, or antibodies which are exposed to the target in a specific region. These leads can be obtained by molecular modeling, which was called as docking, with the help of computer graphics. A combinatorial chemistry plays a major role in this attempt to get the lead, and this technique generates billions of compounds, which was most essential in nowadays in the growing scenario of the diseases. If any compound shows a positive response, traditionally the laboratory methods are there which are used to manufacture them at a large scale. So in this target validation and lead discovery, the biotechnological proteins such as drugs, the use of recombinant DNA technology, genetic engineering to clone and express human genes is another method used to obtain these leads. And then after that lead identification, the next step was lead optimization. The leads are optimized for efficacy and potency in vitro and in vivo, the physical properties, pharmacokinetic properties, and toxicological aspects. And next was preclinical safety. It includes developing a method of large scale of synthesis, animal safety studies, carcinogenetic studies, drug delivery elimination and metabolism studies, drug formulation experiment, and dose ranging studies in animals. So it is most important for every drug to go for the preclinical safety and clinical trials. And the most important one, the next stage of the disease was clinical trials, which the National Institute of Health organizes the clinical trials into 5 different types. One was treatment trial, prevention trial, diagnostic trial, screening trials, and quality of life trial. So let me go for the first one, treatment trial. This trial excludes the new combination of the drugs which are never formed till now, and this new combination plays a major role in the identification and suppression of the disease from the excipients, and also the drug plays a major role. And next was prevention trials. This trial search a new way to prevent a disease or prevent it from recurring. Diagnostic trials. This trial finds better tests or procedure for diagnosing a disease. And next was screening trials. This trial tests methods for detecting diseases, and the last one, quality of life trial. This trial explores ways to improve the comfort and quality of life for individuals with a chronic illness. So these are the five steps involved in the clinical trial development, so that the drug which was released into the market is being safer and effective in treatment of various diseases. Thank you for hearing the stages of drug discovery. Thank you.